degan mavzuga bag'ishlanadi. Bu seminarni ma'ruzachisi Samuel Bickel, Graz texnologiya universitetidan, Avstriyadan. Hozir so'z navbatini Samuel Bickelga beramiz. U o'sha bu mavzu bo'yicha to'liq ma'lumotni prezentatsiyada o'sha ingliz tilida o'sha ma'ruza qiladi. O'zbek tiliga tarjima qilishda o'zim tarjima qilib turaman. Hozir mikrofon yo'q. O'shaning uchun biz mikrofon gapiramiz. Iltimos qilardik, shovqin solmasdan eshitishlaringizni. Rahmat.
Not working.
No. Do we have to? So we do it without the microphone, okay? Yeah, thank you very much for the introduction and for joining us here. Still here. Yeah, as mentioned, I will talk a bit about multi-omics approaches and also how we can use it for microbial preparations, and which means, of course, culturing microorganisms. I didn't know exactly who will be the audience and what you already know and what you don't know. So maybe if it's too boring, say I should go fast, and if it's not, how to say, too complicated, please ask so we can directly discuss. Yeah, so what we do at the Institute of Environmental Biotechnology, we search in nature for functions and things that we can apply. And of course, there's many things in nature we don't understand, so we have to first kind of develop an understanding of what's happening. And then in a second step, we can try to work with those organisms, maybe even entire communities. And in a last step, translate it to some application in agriculture or other industrial applications. And this is our group in Graz. So we are in Austria, in a small town, very small town. This is one picture of our group. It changes all the time, so it's difficult to get a picture with all the members. Here's Gabriele Berg. Some of you may know her from work on the plant microbiome and many other aspects. And she's the head of the institute, so I'm also representing a bit our institute. So we care about these processes, as I mentioned, because the microbes have so many functions. And here is an illustration from a paper review about how microbes in agroecosystems function. And maybe it's not so visible here. What is so nicely shown as cells growing is really hard to observe in nature. So we cannot see them, we cannot open a soil, we don't see where the microbes are and what they are doing. And this is one picture of a small-scale image. So these are clay particles and cells stuck there. But we also know that many of those organisms, we cannot culture them. So we cannot just simply take the soil, extract them, and grow them in the lab. Maybe one question for everyone: how many of you work with environmental microorganisms? Or how many, maybe raise your hand if you work with microbes that you isolate from the environment? Maybe,
Hozir aytib o'tdi o'sha o'zini Graz universiteti haqida, biroz o'sha funksiyalari qanaqa, o'sha laboratoriyasidagi faoliyat haqida aytib o'tdi. Asosan ularning yo'nalishi o'sha tuproq mikrobiologiyasi, agromikrobiologiya, atrof mikrobiologiyasi va asosan o'simliklar mikrobiomi bilan shug'ullanadigan. Hozir bu slaydda o'sha mikrobionda, ya'ni o'sha tashqi muhitda ishlaydigan mikroorganizmlarning asosiy vazifalari va ekosistemani funksiyasi haqida aytib o'tdi. Ya'ni tashqi muhitda, masalan, oddiy tuproq misolida aytib o'tdi, juda ko'p mikroorganizmlar, mikroblar dunyosi mavjud bo'lib, biz bu tuproqda qanaqa mikroorganizmlar borligini oddiy ko'z bilan ko'ra olmaymiz. Ularning ba'zilari masalan o'sha oziqa muhitida ajratib olishni o'stiramiz, ba'zilari oziqa muhitida o'sadi, ba'zilari o'stirish mumkin emas narsani. Va bu mikroorganizmlar o'sha funksiyalarni o'zgarishda to'g'ridan to'g'ri ta'sir etadi va atrof muhitga hamda inson sog'lig'iga o'sha javobgar, ya'ni directly o'sha to'g'ridan to'g'ri ta'sir etadi deb aytib o'tib ketdi. Yana u hozir oxirida savol berdi. o'sha mikrobiom haqida qanaqa tushuncha bor, mikrobiom qanaqa ta'sir etadi, qanaqa funksiyasi bor, shu bo'yicha kimda qanaqa fikr bor deb savol berdim.
Ko'p kulturalar tarjima qiling, ko'p kulturalar ekib bo'lmaydi. Lekin uni обязательно ta'siri bor. Bu tarkibda bo'lish kerak mikrobiomda. O'zining funksiyasi bor, lekin ko'paytirib bo'lmaydi. Shuni yozishga muhtoj.
There are more types of microorganisms which is not able to unculture, but
Yes.
it is, has affected it, but they have their own function and they have their own processes.
Yes, so that's exactly what we observe in nature. All things function, but we cannot figure out how exactly. And this is still where culturing is important. And I will talk a bit about those two approaches we have. One maybe where we can really culture them, and the other one where we rely on other methods to figure out what is happening. And these are mainly multi-omics methods. So it's a lot of bioinformatics, DNA sequencing, RNA sequencing, and so on. Maybe just a question, who of you works with isolates of bacteria on agar plates or in other systems?
Many of them working with isolating microbacteria from soil.
Okay, so many of you work with like this. I was not sure. And how many of you work with bioinformatics? More sequencing?
Yes.
Yeah.
Yeah.
Okay, so we have the both here. So just for me to know what to explain. So the goal of this seminar is to first start some discussion on this topic, and it would be nice if you have questions and comments. And I will try to explain a bit the concept of multi-omics in microbiology and why we need it, and then maybe some very brief mention of culturing techniques or cultivation techniques that maybe try to go a bit beyond what we already do. And then I will mention very briefly some tools that can be used for bioinformatic analysis, but I will not go into the details of the different methods. Yeah. I'm not sure if you want to translate every time?
Okay. Hozirgi mana shu seminarning asosiy maqsadi multi-omikani mikrobiologiyada qanaqa ahamiyati bor va mikrobial o'sha shtammlarni ajratishda qanaqa kultivatsiya texnologiyalarini o'sha kashf etilgan ajratish texnologiyasi va multi-omik ma'lumotlarni analiz qilish uchun o'sha bioinformatik dasturlar haqida bo'ladi asosiy o'sha seminarda olinadigan ma'lumotlar. Qanaqa savollaringiz bo'lsa, qanaqa o'sha joyga tushunmagan bo'lsalaringiz, hozir oramizda menimcha ko'pchilik ingliz tilini yaxshi tushunadi. Bu anaqa nima? Seminarda ham mavzu qanday ma'noda ketayotganini tushunib, ingliz tilida ham bemalol qatnashishlaringiz mumkin, savol berishlaringiz mumkin. Aktiv qatnashishlaringizni so'radim.
So the overall approach is a bit illustrated here. So what we're trying to do is we want to learn what nature already does, analyze the data, look at what is the underlying mechanisms, and then try to predict it. Of course, then we have to test it. So for that, we often have to see if those organisms actually have this function, and then we update our thinking and go again. Okay. Okay. Let's stop. Yes. Yes. Yes.
Bekorga vaqt oladi, ko'pchilik tushunadi. Tushunmaganni so'raydi. Aytsoqa, ko'pchilik bu prezentatsiyaga qiziq tushunishi shunaqa qildi desangiz xafa bo'lmaydi.
Kerak jo'ydi. O'tiringlar, boshlaylik. Please, continue.
Hozir bitta narsani sizlardan so'ramoqchi edim. Bu agar to'g'ridan to'g'ri har bir gapni tarjima qiladigan bo'lsak, o'zi anaqasi uzun ekan, seminar anaqa prezentatsiyasi. To'g'ridan to'g'ri direct anaqa qilish, muloqot qilishni xohlayapti, chunki baribir ko'pchilik oramizda ingliz tilini yaxshi tushunadi-ku, to'g'rimi? Oddiy tilda, akademik til emas, oddiyroq tilda gapiryapti. Agar o'shaning uchun hozir taklif qilyapti, tushunmagan joylaringiz bo'lsa, tarjima qilsam, bu davom etib bemalol sizlar bilan ingliz tilida ishlasa. Nima deysizlar?
Bo'ladi.
Понимаем, но не говорим. Можно, да, остаться?
Да. No.
So, yeah. So as I mentioned, I will try to introduce some of the ideas and concepts for this type of analysis. And maybe the important part is to remember also, usually it's not one method that is enough to characterize a microbiome. So there's so many factors and so many processes that it's really difficult to, I don't know, just do culture only or do sequencing only. We need a combination of methods to really understand what's happening. So very broadly, we can categorize the approaches into two methods. So one, the culture-dependent method, where we can use different media to select different organisms or grow with different nutrients and so on. Or we have the culture-independent methods, where we can also capture microorganisms that are not culturable. And we know that only very few are cultured. So here I wrote 1%, but actually it's maybe a bit more. I don't know, the number changes every year. It can be 1% up to 10%, depending on the literature. But I would say about 1 to 2% is maybe a good estimate. And we know that it is not a problem that those organisms don't grow, we just don't know what they need. So it's often the problem that we don't know what media is needed for cultivation. And sometimes it's more complicated than just the media, because they rely on other microbes. And this is very challenging, because we have to suddenly cultivate two, because they need each other to grow. And this, I will also not go into detail, but this is a very challenging thing to first figure out and then apply also. Then the benefit of culture-dependent methods is really that we can develop a culture collection. So we can keep the isolates, we can store them. If we need them again, we can get them out and grow them again. So this also offers the way for biotechnological applications. Because if we cannot culture them, we cannot grow them in a bioreactor, and we cannot apply them in different fields, for example, to harvest enzymes or to apply them to the field as bioinoculants. This is only possible if we can grow them. The culture-independent methods, on the other hand, they provide a lot of information. Sometimes maybe too much information to keep an overview, but they provide a lot of information, so we can know which organisms are there, what kind of metabolic activity they could have, so what genes are present, and this allows us to describe also multi-species system. It is a bit more expensive, likely, because in addition to the cost of work, of labor, it's also the method itself is a bit more expensive, but then also the analysis of the data is a lot more expensive because it takes much more time to analyze these large data sets than to analyze more simple culture-based methods. This is just something to keep in mind, and it might also change. So depending on how automated the system is, these costs can also change. Now, I mentioned that many are not culturable, and this is a graph showing how many sequences are deposited in, I think it was NCBI, so people sequence something and they find, oh, this is interesting, and they deposit it in the database, and this has been growing over the years. And here is the cumulative figure, so it's up to 700,000 sequences that are deposited, so there's a lot to analyze.
Yes.
It is really rapidly growing, so these are numbers from 2018, so it's already almost 10 years ago, but you can see that how the accumulation of this information is still increasing. So there's many, many more sequences that are found every day, probably, and yeah, it's all information that is there that we, at the end, are not entirely sure what it means. Because again, this is a paper from just this September, where they looked at also culturable and unculturable methods, and again they cite 2% are culturable. So these are those where we can validate their function. We can grow them, apply the conditions, and test if they can do a certain functions. For the others, we rely on sequencing data, which is a potential or a gene, and we are not sure if it's really expressed or it's really promoting this function. And in this paper, they they sequenced more or less one soil, and they sequenced it really, really deep with a lot of sequencing information. And now, yeah, it's at the terabase scale, so this is quite a big scale. If we think of one genome has maybe 1 gigabase, roughly. So this would be about still only 1,000 genomes. So we are still far away from really characterizing the many thousands of species that could live in a soil. Yeah, this is just one example, and it's interesting because it did not change so much since the last 10 years. So still, the technologies are better, we get more and more data, but we can still only culture a small, tiny fraction. Then why should we then need the multi-omics for this kind of microbial preparations and to work with them? Because you could also say, well, we don't know what they do and we cannot validate it, why should we collect all this data? It's still really important because, for example, just amplicon sequencing alone, so this is a 16S marker gene that allows us to distinguish the taxonomy. Even this alone is very useful to estimate how many species are there and how they are related to each other. And there are estimates that there are around 400,000 different species, which are then associated to about 60,000 genera. So this is, again, a big jump from, this means that's about almost 7 times more species than we have genera. So it really expands the number of possibilities we have and the number of taxa that we can find. And from those, there's only around 14,000 archaea and bacteria that are actually cultivated on their own and can be described and validated. So this really shows that while we find a lot of things, we can really only fully study a small fraction. And if we then look even further, so these 14,000, they are distributed on 3,500 genera. So you can see the difference between 60,000 genera that are potentially there and 3,500 that we can actually culture. So there is a big gap. And to make things even a bit more challenging, many of the cultured ones are biased to a few groups like Bacteroideotes, Proteobacteria, Firmicutes, and Actinobacteria. For many other groups, we even have much more difficulties to culture. And I think everyone who knows growing a bacteria in the lab, they know the difficulties that small changes in the media or the lack of a certain compound, nothing grows anymore. And sometimes things grow and you don't know why. So it's also, it can go both ways. And this is really, I think, summarized also in this fact that we know there should be many, many more, but we can actually only really culture this small fraction. And it is selected only a few groups of bacteria. We cannot even say, okay, we have a representative sample of all the culturable ones. And then the question is, okay, we can sequence it and we can assign functions and so on. Why do we still need to culture? I think I mentioned several things already. Once, of course, we can create pure cultures, so we can remove this one organism from the whole system and study it alone. So this allows us to replicate studies and also to improve the repeatability and the statistics, because you can expose this one organism to different conditions and so on. However, we also lose the context. So we lose the associations this organism has potentially with others. And nonetheless, it's very difficult to assess certain traits just from the genome itself. One example is motility, bacterial motility. We know there are some factors and proteins that are needed to make a flagellum and swim, but even the existence of the motor protein and the flagellum does not mean that the organism can actually swim. And under what conditions it will swim, we also don't know. So this is one example where it's very challenging, because nature is very inventive in also using the same system for multiple purposes. For example, the flagellar proteins, they can also be used to inject things into other cells and extend nanotubes and things like that. So just by having the gene for making a component of the flagella doesn't mean it is actually motile. And this is one example where it's very challenging just based on the genome to understand what the organism is really doing. And yeah, as I mentioned, sometimes we need also a high biomass. So when we culture them, we can reach much higher numbers, and then we also get a better analysis of the genomes, for example. This is not easy to recover from a natural system where there's a 1 in 1,000 species and you try to find this one genome. It's very challenging. Yeah, I think, yeah, if you have questions about this, please ask. Otherwise, we'll continue. Nonetheless, the culturing is also crucial to find new, as I mentioned, new approaches for, in this case, for example, sustainable agriculture. Because even if we can sequence those and we can characterize them, at the end we cannot apply them. And that's what we want to do. No? We want to take the microbes and we can figure out what they need. This is also sometimes termed culture-omics. So this is a high throughput culturing where you just apply many, many conditions and screen all the possibilities. So we can also culture organisms that maybe are thought to be unculturable by just searching for different conditions. And at the end, we can grow them in a bioreactor and apply them to the field. So this is really the core also of why culturing will never disappear. I think it's really an essential part of microbiology and also the application of it, that we can have the organism itself and describe it properly and validate the functions. And of course this also led to an increase in techniques that are used for culturing. And some of them are listed here, so this is a small summary. And these are mainly strategies to culture environmental bacteria. One example is simply use media with less nutrients. So this is a simple trick and is very successful for many strains. They get outcompeted, but then with less nutrients, suddenly they grow and we can isolate them. Or simulating the natural environment. So there's different approaches to do that. For example, in microbial chambers, or some use even leaves itself as a substrate to grow bacteria on, or fungi. Or maybe a more controlled approach is to use microfluidic technologies, where you can apply gradients of nutrients at a very small scale and see which cells accumulate in which location and then re-isolate them from there. Another approach which relies more on equipment is single cell separation, for example with a cell sorter. So you can then take a natural sample and sort the cells and extract, for example, only a certain clade or certain active microbes. But this also works with dilution to extinction culturing, where you just, as you know, dilute until you have a pure culture of different microbes. Again, this works only if you know what conditions they need to grow. When it becomes really tricky with culturing is cultivation in co-culture. So if you know this organism relies on another one, and then you have to grow them together. So often when you have a liquid culture, you grow them together, only one survives, the other just kind of disappears. And this is very challenging to find these conditions which enable both to grow. And the other challenge is the cultivation of slow-growing bacteria, because I think most of us will not have the time. A PhD is 3, 4, 5 years, but if the bacterium doubles once every half a year, then yeah, you need a lot of time. And this is also a reason for the bias observed in the culturable, because we like to work fast. We like to have them overnight and have a colony, and then we can continue the work. We don't want to wait 3 months just to see 10 cells, and yeah. So it's very challenging, and it's also an issue of how the science is organized and financed. So this is maybe less of a problem of the microorganisms, but also the challenge to conduct long-term studies. And of course, for biotechnological applications, it's also challenging if the microbe grows so slow, because it costs more, it takes more time, there's more risk for contaminations, and so on. Yeah.
It is very difficult.
It's very difficult, yes. So, to sum this up, there are innovations in culturing, but at the end, the basics are the same. Here some are listed, so some are like motility-based enrichment, so if you know your organism is motile, there's ways to target those. Selective media, or separation based on density or on size, so using different filters to get different sizes of bacteria. Then also, of course, selective inhibitors, we know from like antibiotics or other compounds. And screening different conditions. But this again has some limits, because we cannot change the temperature however we want in a lab. It's also difficult, and pH might be a bit easier. Then, as I mentioned, dilution to extinction and basic colony picking, which is still, I think, one of the most used culturing or isolation techniques. And the challenges are really to identify the substrates and control the growth, figure out if some need physical contact or spatial proximity, so they need to be close to another organism, and also sometimes they need to be attached to surfaces to really grow, so this is also another challenging thing, especially in liquid cultures. And some may just be sleeping, so we don't know how to wake them up. And this is again can be a bit of a stochastic process, so randomly one wakes up or not, so we don't know what exactly controls the dormancy. But it's possible you can plate them and they just don't grow because they don't want to. And then you have to wait, and maybe one wakes up and then you're lucky, but it's really tricky to know what are the triggers of dormancy. So, and I mentioned this kind of symbiotic associations, which is often, for example, for host-associated cells, which rely on close contact to a plant or to another organism. This is also difficult to then do in the lab. And this is maybe where multi-omics comes. So, what is multi-omics? It's a bit of a strange term at the end. It's just a summary of all the terms that end with omes, which is genome, epigenome, transcriptome, proteome, metabolome, exposome, microbiome. All of those terms together are considered omics. And multi-omics techniques are used in all the different parts here. So genome, we know it's the DNA content of an organism. Epigenome is already very challenging. What is the modification of this genome? Transcriptome, which genes are expressed as a whole, so in the whole culture. Then proteome, which proteins are expressed or translated. And then the metabolome, what kind of metabolites are produced or consumed during this process. The exposome is a term that comes from chemistry. This is the total of exposures an organism is experiencing. So for example, to do, especially at the extraction stage. If you have to extract RNA from a sample, it's really hard because RNA degrades within 2 minutes, 3 minutes, and then it's gone or degraded in a bad quality, and then it's very challenging to get good sequencing data. While DNA can last for maybe half a month or so without much problem. I mean, it also degrades, but it's still available and you can still get information out of it. So for RNA extraction is really crucial because it needs the whole approach, the whole method needs to be also trained. You cannot do it in the first time. You need to optimize every step. Samples need to be cooled and every component needs to be ready at the correct time. And this is just the handling of the extraction. So this is already challenging. What's also challenging with the RNAs that you need a good reference. You need a good reference condition. So you need a control which you know that the only thing that I'm changing is the condition of interest. Because many genes are up and down regulated and it's very noisy also. The organisms express sometimes randomly certain genes and so on. So if you don't have a good reference, it's really hard to analyze the data. And this is important because this starts already at the experimental design. You can have the best RNA extraction and really good sequencing data. If you don't know what to compare it against, it's more or less useless. It's very hard. It's really hard to draw inferences or to understand what's going on from RNA if you don't have a good reference. So you need a very good control. This can be a treatment, this can be, for example, if you, let's say you have a media and you add glucose. You cannot use as a control another media or water or something. It will change so many genes, it will not be comparable at all. So it needs to be really well defined. Maybe also what are you trying to compare and what is the information you want to gain. Yeah, and the approach is very similar. So we have just a messenger RNA that is from the expressed genes in the sample. Maybe you apply some stress to the sample to induce the change, and then you compare it against the unstressed sample. The approach is otherwise similar. It just at the end, mRNA will first be extracted and then transformed often to cDNA to have it stable, and then it's sequenced like the other methods. So after the extraction and the conversion to DNA, there is no difference in the method in itself. In the data analysis, there is some different algorithms to use, but overall, I think it's very comparable from the sequencing point of view. And the good thing here is it gives us not only taxonomic information, but also which genes are expressed. So we can see who is doing what, essentially, or trying to do what, to be very correct. Yeah. So what are the benefits? Shotgun metagenomics or transcriptomics, you can recover entire genome sequences from entire communities, not only selected organisms or specific markers. So it gives us a very broad picture. Then this allows us to analyze or investigate complex microbial populations at a larger scale. I mean, large for the scale of microbes. So, for example, a soil sample of 1 gram is already huge with so many microbes inside. It allows us who is there, who is active, and it gives us the metabolic potential or the intention of the organisms to do something. Again, as I mentioned, sample handling is more crucial than with other approaches, such as amplicons, for example. And it is a bit more costly, because you have to sequence much deeper. Then another issue is the host sequence. So we have DNA from the host. This can be a plant, insects, or humans, or animals. And this also makes an issue with the sequencing, because many sequences will not be bacterial. If we are interested in bacteria and we have a plant host, we will get much DNA from the plant. So this is also important already at the step of extraction and how to treat the samples to minimize the host DNA. Unless you're interested in the host DNA, then of course there's no way around increasing sequencing depth.
Emmanuel, the shotgun,
yes?
It's sequencing of some genes in genome, yes?
No,
Not
it's
all.
any genes.
Any genes?
Yes. It's just whatever gene fragment is there.
For example, if we mix two strains of one species, can we separate it? You know?
Yeah, that's. Yeah, that's what.
We saw
I think it depends if you have a template or so. If you can grow them individually, you can first sequence each of the genomes, hope that they are consistent, and then once you mix them, maybe you can. But no, most likely, if you have a big overlap, you will not be able to really attribute it. I think for this kind of situations, you probably need something like isotope labeling. So you can give it a heavy, for example, a heavy carbon source that only one of them can use. So then you can do density fractionation maybe, and separate it this way. But this is a very challenging.
Yes. No.
Yeah. Yeah, if they're closely related, same species, I think strain level will be very challenging.
Yes.
Especially considering that some species have a kind of a pangenome and a lot of flexibility in their genes. So I think, for example, Bacillus is one of those as well. Subtilis, they can incorporate DNA from the environment, and then it gets very challenging for them.
That's true.
Yeah. But maybe, no, I think that's even with better computational methods, I don't think this is something that can be solved. It's the nature of it. In this case, what would be probably the best is sequence the genomes individually and then do a differential analysis to see which genes are present in one and not in the other, and then target only those genes for the expression. For example, get the primer for those genes and do a marker gene analysis on those genes. That could be, I think, the best way in this case. Because then you remove all the other genes and focus only on the ones that you know are different between the two organisms. But this is a case where you already culture them, so you know what's happening. In the natural community, we just call them genus or species. I don't think we can go further than that. Yes, and a last thing here, what is also a limitation, that many computational methods, they rely on already accessible genomes. So the database is there and we compare against this database. So if it's not in the database, we might assign it to some other organism, or we don't know what it is. And this is, of course, a problem because, as we know, these databases are mainly made up of culturable organisms. At least the ones with a high quality genomes and so on, they are typically from cultured organisms. So there's some bias in the database also to not only organisms or groups of organisms, but also to environments. So many databases are heavily influenced by clinical samples, a lot less from other environments. For example, plant endophytes are very challenging to find in those databases and so on. So there's also an inherent bias in this method because we rely on databases for genome assembly, genome annotation, for all those things. Of course, it depends on the algorithms. So this is also going to this direction. Why do we still do amplicon sequencing if the other one is much better? And I think one of the examples is, of course, the cost, which is much lower for amplicons. The sample preparation is easier. We get a lot of DNA. We do a PCR, so we amplify whatever is there. We don't have the problem that we don't have enough DNA, and sometimes still, but a lot less. The taxonomic resolution can be higher, and especially the how wide we can see the taxonomy is higher. So we can capture more of the diversity by amplicons than by metagenomes. Just simply because the number of sequences is limited. So either we have to invest much more money and sequence much deeper, or we cannot capture the full diversity. So as an example from soil, for a typical metagenome, maybe we can assemble 100 genomes from different taxa, maybe a bit more. For amplicons, we can recover 1,000 or 10,000 ASVs or OTUs. So there's a big difference in characterizing the diversity. Of course, you can argue they are much lower in abundance, so they are maybe not the most dominant ones for the functions, but this is open for discussion. I think sometimes we're interested in the less abundant taxa. So there's some enrichment will be needed to really capture those with the metagenomes. Yeah, so overall, and as I mentioned, the bioinformatics requirements are much lower. So it takes less computational power. You can probably run most of the analysis, if it's not too large a data set, on your laptop, while for metagenomes, this will be challenging. So to assemble a genome or analyze a genome, it quickly takes tens of minutes or half an hour, and for amplicons you can run it typically on smaller computers. Yeah, and it still allows us to see what is happening in the microbial community. So it's not, and we are not limited to 16S. We can use 16S, but we can use other marker genes as well. So even there, it has some benefit that if we know what we're looking for, we can target it and amplify it and analyze it much easier than going for full metagenomics. However, if we want to see the whole community's capacity on the metabolism and maybe try to infer pathways and so on, then we need metagenomics. And at the end, if we want to know the community activity, we need to consider RNA or metatranscriptomics or even metabolomics. So we want to then really analyze the metabolites that are produced or consumed. And again, it's usually multiple approaches that we need to really understand what is happening in the microbiome. It's not one method for everything. Yes, so this is just one slide to maybe summarize very few tools. I know this is very short, but of course we can, if questions, we can discuss. I guess you can also get the slides, so you have the links to those. Here I just put CHIME tool for bioinformatics analysis. I guess some are familiar with this also. Because it is a very well developed platform, so essentially it's not only one tool, it's a wrapper for many different tools, so it provides an interface to many of the tools that are available, and it's very well tested, and it has kind of a standard workflow, let's say. Recently, I think only a year ago, they have a second version also for metagenomic data. So this is why it's, to me, very nice to recommend CHIME, especially if you start with this kind of work, because it has many tutorials, first of all, so you can really go to the web page. They have data sets you can try out. There's tutorials. So I think especially for students and for maybe master students, it's a bit, they don't have enough time to really work with it unless it's the bioinformatics master. But for PhD students, I think it's very suitable to really just dig through the documentation and look at the tutorials that are there.
That's free?
It's free. Yes. That's maybe I have to mention. All the software I mentioned here is open source and free. So you can just download it and install it and run it without any cost. I think, apart from the computational, if you need a server or something, or a powerful computer, but nowadays I think many laptops are powerful enough to run amplicon data without a problem. I think this is a very nice thing.
Good for students.
Very good for students, I think. And for research, because we don't pay licenses for this, and also it is well documented. People can ask questions online and the developers answer, so it's really, there's a lot of content there where you can, it's also transparent what is happening. It's not a company that hides the algorithm behind some closed source code, some app. It's really an open thing, so you can look at every code if you want. And then for general data analysis, for me, Python is very nice. I mean, here I mentioned a few packages. Python is a programming language. Actually, a lot of CHIME in the background is also implemented in Python. So if you want to go deeper into what is happening in the code, it is very useful to understand programming language, such as Python or R. Both are used heavily also in CHIME. And these are maybe Yeah, so the learning curve here is a bit steeper, I think, because you need to understand how programs are built, how data is input, how you transform data, but it can be very helpful to develop this kind of analysis. And for example, also visualization of data. There's a nice package in Python called Seaborn. It allows you to say, plot me a box plot with this and this condition, and so on. So it's very, very useful, but you need to learn a bit how to code. Again, there's many tutorials. So also on their webpage, they have a gallery where they show the different types of graphs. You can click on it and you can already see what code they used to make it. So this is also very, very helpful to get started. And to get some ideas also, how could I analyze the data? The same with R, there's a lot of packages. Two famous ones, Philosec is very useful to load microbiome data, so they can handle the different types of data for microbiomes. Typically there is one table with ASV and samples, so it has the number of ASVs and the number of samples, and then it tells you how many times it was detected in one sample, so how many reads you have. The second table is usually the taxonomic table, which tells you this ASV or this OTU can be assigned to this taxonomy, so this genus. And a third table usually describes your experimental conditions, which gives you more information about the samples. So for example, what were the treatments, what were the temperatures, and so on. And it's typically these three data inputs that are needed for any kind of microbiome analysis. And especially Philosec, it takes some more input. It also can include the taxonomic tree, so the phylogenetic tree. But yeah, so this is mainly the type of data that is then used in these programs. Yeah. For Python, there is something called Anaconda. It's also free. You can download it. The good thing there is it already comes with the most important packages, and it's also consistent. So all the versioning, everything should work out of the box. So you can download it, install it, and start working with it. Same with RStudio. It already comes with many functions that are needed. Yeah. So if you have questions to this, maybe we can discuss. Otherwise, I will just show very few examples of where we did some research where we used these kind of techniques. I mean, very basic ones. I'm not going to too much details. So one was a project called Excalibur that was about bioinoculants. And here you can see just an example where we had a field which was treated with verticillium. And these are the plates that were recovered from the field treated with verticillium and those without. And this is verticillium growing. So in this case it was very easy. Simple culturing was enough to just test if they successfully colonized the soil. So this is really an example where they applied a bioinoculant in the field. And they went out, took the soil and just plated it, and then you find it again. I think this is a very rare case, but it can work. And if it works, then yeah, I think if you can validate that this is indeed the strain that you applied, then it's good enough. You don't need to do all the other complicated analysis to see if it's successfully colonized. Another example that we did was we had small microcosms where we had volatiles and bioinoculants. So we put the bacterium that is supposed to produce some volatiles, and then we did amplicon sequencing. So I forgot to add up here is bacteria and down here fungi. Sorry, and these are two different soils. So in the triangles here are one soil type, and the circles are another soil type. What we observed there was that first the bacterial community. So if the points are closer together, the communities are more similar. So if we apply the treatment, some bacterial communities reacted, but not so clearly. For fungi, we saw a big change. So the fungal community responded very strongly to this treatment, and only in one of the soils. So this was interesting because this was a soil from the field, and the circle is a soil from potting mix for a garden soil, like a horticultural soil, and this one had a very strong response. And then we used the same soil, so the same extract, and we used phenotyping. So this is a way to see which carbon sources can be utilized by the microorganisms there. And what we saw that there are so this is the utilization rate of the carbon source in the control treatment, and this is the one where we had the bioinoculant. And we just see some of them are increasingly used if we provide our inoculant. And this is also shown here, the same thing. So for example, as an example, glucose 1-phosphate was increasingly utilized after we apply a bioinoculant. Now, of course, we don't have the direct link, but we can speculate that this change in the utilization pattern was mainly driven by the fungi, because that's indicated from the amplicons. So there's two things we see from the marker genes. There's a shift in the community, and we see from the phenotypic assessment there is a change in the carbon sources that are used. So we can, of course, we don't have the full proof. For that we need RNA and everything, but it gives us a good idea that likely it was the fungi that were affected, mainly driving this change. And interestingly, we saw it only in one of the soil types again. So it mirrors the response in the metabolism mirrors the response in the amplicons. So also here in the same soil type, they didn't respond. The community didn't change. The metabolism for the community also didn't change. But in the other soil, we saw the change in the community and in the metabolism. So this gives us a good indication that there's actually something really happening from our inoculants. It's not just new DNA that was added. It's also affecting the composition of the community that then affects the whole metabolism of the community. Another paper was about characterizing Penibacillus polymixa. So this is a well-known strain used also in agriculture. And it has many effects for plant nutrition and the growth of plants and plant health. And what we did again was just simple screening for the phenotype. So we saw which carbon source, it's very small, I'm sorry. In blue is sulfur sources, in N is nitrogen sources, in black is carbon sources. And we just saw there are certain carbon sources that are used and others not. This can be then combined with the genome data, which is here shown as a KEC pathway. And here you can see which of those sources were used. So a very strong purple color shows that this source, in this case an amino acid, was used increasingly by this organism. And we can map this to the gene, to the genes also. Because these are the KEC IDs for the EC numbers for the enzymes. So each of those reactions can be mapped to a gene. And this is a simple way to combine this information because it only required a genome and the addition of a phenotypic assessment. In this case, these are microarrays which have many different carbon sources where you can just check how well they use them or if they use them or not. In addition, we also measured the volatiles, so gases that are produced, and we found several new gases that were not documented for this organism. For example, pyrazine in red, which are known to have also antifungal activity. So this was very interesting because this was not obvious from the genome annotation, but when we then grow the culture and collect the gases that come out with GCMS, we saw, okay, they're actually also producing this. We didn't go further, so this is, again, you can go much further into details, but it just shows it's very helpful to combine these techniques because some properties are missed in the genome. Another thing that we found also from the genome was antimicrobial resistances. So in the genome it pops up, oh, they have a lot of antimicrobial resistances. And this is of course a problem if we want to apply this strain everywhere on fields and so on. So we were thinking, this is not good, and we should test it. They actually tested it then in the lab, and it shows very, very low resistance. So while the gene was flagging, careful, careful, this is potentially dangerous, when we then exposed them to the antibiotics, they were almost none were really resistant. So we don't really know why this is. It could be that the annotation of the gene was not correct. It could also be that this gene is not expressed so strongly, or the conditions were not met. But it just shows that alone on the genome, it's very hard to interpret this. And it's also a risk for regulatory purposes, because I know in the EU there is some ideas to have all the bioinoculants that would enter the market, they should annotate the genomes and deposit to the government. And then they look at it and say, oh no, this is too risky, this is dangerous, and so on. And this is a very critical practice. And I think it's important to remember that just knowing the genome will not tell us what it means. Although it would be easy for government to have a list, oh, this is dangerous, this and this gene, so if you find it, no, you cannot use it. Which would limit us a lot. So I hope this will not go through. There's a lot of discussions and a lot of commentaries by different groups going against these kind of ideas, but it is there. Because the idea is more data, we know more, so we can just regulate it this way. And this is maybe also important for the discussion, I think. Maybe I will not say much here. So this is a project we are working on together with, led by Licio Malusa from Poland. He is, in here we are trying to develop new substrates for horticulture. And we are also trying to combine again these approaches. So here you can see different soils and substrates, and we analyze their gas emissions, because we know they are critical for also fending off pathogens. In this case, we did an experiment with Rhizoctonia solani and tomato plants. So we want to really figure out what that makes a soil good for growing plants, what makes disease-suppressive soils, and these kind of things. And I don't have data yet, so I will just show the general idea, and the approach we're taking is again, maybe can be called a multi-omics approach in that sense. We want to screen different substrates and soils. We extract the cells for isolation and preserving them. So it's simple culturing with different media. We will do some functional phenotyping. At the same time, we extract the DNA. We'll do qPCR and metagenomic sequencing to, at the end, establish a database. And this database will be also combined with the volatiles, so the gas that is emitted from this. So this is a lot of work, and it needs a lot of coordination, because we need to make sure that the samples are treated the same way for the different analysis. But at the end, we hopefully will have a database which tells us, okay, this substrate has this bacteria inside, they have certain functions that can be useful. Then we can also show that those functions are actually working, so we have the isolates, we can go back and check. And then the hope is that we can use this information to develop a new substrate. Amazing. So we want to combine it in the way that we get the best, for example, kind of a personalized substrate for different plants. So this is the idea, and I think something like this is not possible with only one technique, and it takes a combination. And as I mentioned earlier, it needs the coordinated effort. So all those steps need to be coordinated, so at the end what we get is really comparable, the data. Yeah, with that I want to thank our team at TU, and of course the microorganisms that are doing most of the work, like some bacteria and fungi growing in the soil. And yeah, if you have questions, I'll be happy to answer.
O'zlari qiziqqan tushunmagan joylarga bemalol so'rasalaringiz bo'ladi.
Quantitative insight into microbial ecology software program,
Sorry?
This program, quantitative insight into microbial ecology software program.
Yes.
Is it possible to accurately identify probiotic strains using, or is it possible to distinguish beneficial
probiotics. Now the question is also probiotics for what? So for plants or for humans or animals? So there you need to first know what kind of functions you're searching for and if they can actually survive in the gut. I guess it's for the gut or... yes, so that's also the question, what part of the animal are you trying to get the probiotics in? Because, for example, it can be in the skin or in oral or in the gut. So I guess maybe the gut. And then the question is, can they survive these conditions? And I don't think that the genomic data itself is sufficient to know this. But what it can help is, if you know what organisms can survive in the gut, you can compare those to your organisms and potentially find an overlap. So you can see what functions do those have in a healthy animal that are maybe not there in a diseased animal. So these kind of comparisons you can do. For example, if you have, let's say you have a cow that is healthy and you have a cow that is unhealthy, and you compare those two compositions of their gut microbiome. This can give you some indication of what microbes are missing in the unhealthy cow. And this way, maybe you can figure out what functions they have and how to supply them. But it's still challenging because you have to, there's a few steps to validate it. Because often, also for human medicine, we don't know, is it the disease causing the change in the microbial composition, or is it the microbial composition causing the disease? And this question cannot be answered just by bioinformatics. It's still useful because you can get an overview over what changes. And then, of course, you would have to go further and maybe identify a function. And the simplest function is the microbe produces something that is healthy for the animal, let's say. And then, of course, you can try to validate it by providing the pure compound and providing the microbe, and then you can see if it's really the microorganisms that is altering the health of the animal.
Maybe this program is Republic Uzbekistan correctly?
The program?
Software.
Yes.
Software program, you see?
Yes. I think definitely it's useful for this kind of investigation. It's still critical is still how the experiment is designed. What is the control group? What is the treatment group? And then, but it's definitely useful for the analysis of this kind of data. And it can give you insight in what is changing between the unhealthy one and the healthy one. So, for example, so let's say you have an animal, you give it this probiotic, and then you take a sample from the gut, and you have another one where you give a placebo, or like a you don't give the probiotic, and then you can compare what changed. I think for this kind of approach it's very useful. And it's probably useful to combine it then also with a metabolic analysis. If you know there's a certain compound that is helpful for the well-being of the animal, then you can search for this. You can search, is the microbe that I have, do they produce this? Or do they have the potential to produce it? Mm-hmm.
Thank you very much.
Yes, you're welcome.
Professor, thank you for your presentation. It is very interesting because it is a multi-omics very important for us. I have only advice for you because we are analyzing microbiome, some different disease. And what do you think it is our program in Python, which is the easier for us? It's the best.
Again, I think there's not one that can solve everything. Personally, for me, Python is easier, especially to handle data, to transform tables, to work with the data. To me, it's much easier. But I learned both. I started with R, and then I later discovered Python. So for me, it is easier to write the code and to read it also. But for certain algorithms and statistics especially has much more developed packages. So there is it's again a balance. What I often do, I arrange my data in Python, group it, I don't know, transform it and so on. Then I export it, run it in R for the statistics, and then I import it back in Python for data visualization. But that's my personal preference also. I heard many people in our group prefer R, also for data visualization. It also has lots of packages. It's not limited at all in that. But to me it's somehow easier. So this is, I think it's a preference at the end. But it's also what I want to, maybe the message I want to say is you can also combine them easily. Often you can export the data, read it in with another program. There is no strict rule which one should be used or is better. What is important is once you know how to program a little bit in one language, it's much easier to learn the other one as well. There is of course some differences that you have to learn, like learning a new language, but it's not like learning an entire new language, it's more like learning another dialect. Unless you take some extreme, like C or something. But yeah, even there, the language is almost the same. Yeah.
Thank you.
I have a question.
Yes?
Can I give a question regarding to health sector?
Yes, I might not be the expert, but...
For example, when we use the PCR tests for COVID, for detecting COVID, these tests, with the help of these tests, maybe identify some mutation and other mutations are not identified. So for screening methods, it was better to use these immunological methods like antibody. For example, for identification of antibiotic-resistant genes, which one is more validated? Identifying these transcriptomes or genome or culturing method, which one is more validated?
I think it's a very important question. I have to say I'm not an expert on the health issues, but I understand the difficulties with PCR, for example. If the thing mutates too much, you will not be able to detect it, or the quantitative PCR will perform very badly. Just the melting curves will look random. And the other thing is, you mentioned the immune response. I think you're measuring a different thing. No? You're measuring what the host is doing and not what the organism is doing.
But know that can we use this PCR test for identification of antibiotic resistance?
Okay. If you know the gene, yes. But the tricky thing there is, I think, especially with natural isolates, what we find is we get a lot of antibiotic resistance genes that we at the end are not entirely sure, is it really an antibiotic resistance or is it just some general transporter or some metabolic gene that, I don't know, for example, plant-associated bacteria, they often show a lot of antibiotic resistance genes. So we find that many of them if we assign them in the database, but then we always wonder, like, why? Because there's not, like endophytes, for example. There's not much growth, there's not much competition, and so on. Why do they need all those genes? And one possibility could be that those genes are just an evolutionary adaptation to the environment. So the plant has a lot of secondary metabolites, and many of them are similar to antibiotics. Maybe some even have antimicrobial properties. But at the end, we are not entirely sure if this is a targeted antibiotic or it's just a general response to these compounds. And with that, what I mean is it's very challenging to say everything that appears as an antibiotic resistant gene is actually only the purpose to defend an antibiotic. Because often they have multiple functions, like a efflux pump, for example. They can export anything that is harmful for the cell. So it's not specific to the antibiotic. But nonetheless, it can be a good indicator and say, oh, careful, this could be resistant. But then I think it's important to test it on the plate. So I would, again, maybe use multiple methods. First, you can do a PCR and see, oh, the gene is here, careful. and then you put it on the plate, expose it to the antibiotic, and test if it's really resistant.
Thank you.
I think that is a good combination.
Thank you very much.
Any question? Savollar bormi? Manda savol bor.
I ask in Uzbek, I will ask Aziza, she will translate because this question many interesting for us who is working in agriculture, microbiology. So, men savolim shunday iboratki, mana metagenomikani o'rganyapti tuproqda. Metagenomikada tuproqdagi o'sha misol uchun bug'doy ekilgan bo'lsa, bug'doyni rizosfera atrofida faqatgina bir xildagi bakteriyalar har vaqt paydo bo'ladimi? Yoki bu o'sha tuproqning xususiyatiga bog'liqmi? Yoki bu fasllar o'zgarishiga bog'liqmi? Mana shu haqida fikrlari bormi? For example,
the last one, sorry, why does the soil appear?
Yes, it's why the soil, the same soil, we will need part of formal.
Okay, so all the diversity in soil.
for example, biz birinchi yili bug'doy ekamiz, ikkinchi yili paxta ekamiz. Metagenomika o'sha tuproqdagi bakteriyalar miqdori o'zgaradimi yo'qmi? Lidlari, turlari o'zgarib ketadimi yo'qmi?
Yeah, so thank you for the question. I think
This is very easy question.
very interesting, and very challenging question, because I think the difficulty here is there's so many factors that play in an agricultural setting. So first, maybe the ones we cannot control, the weather. So it's the same soil, but one year is wet, the other year is dry, hot or cold. It all alters the community. And then the next is, of course, the crop and the way the crop is managed. So, for example, maybe wheat has more demand for, maybe less demand for water than cotton. So already there it has an interaction with the climate and with the soil and with the plant. But then also each plant has its own microbiome. So each plant has endophytes also, they bring already to the field. And then each plant selects differently from the environment. So the roots, they release carbon to the soil, microbes come, and already there's a different enrichment of organisms. So this also changes the community. To make it even more complicated, this changes over the season, because the plant changes over the season. It starts very small, maybe the influence on the soil is not so big. Then it gets bigger, it influences more of the soil, then the plant changes its condition, it's flowering, maybe it takes up a lot of phosphate, it uses different nutrients, and then it turns ripe and so on. So even there, the plant developmental stage also changes the composition of the microbiome. And there we know very little about, actually, how the microbiome changes over time for the plants. Of course, in the field this is difficult to study because the weather and everything also changes. Yes. And then what comes on top of it even is the agricultural management. So different crops are managed differently. So sometimes pesticides or herbicides are used, fungicides are a very, very impactful thing. And we had this case as well in some studies where colleagues, they were sampling and so on, and they were trying to extract DNA and then analyzing it and so on. And why they know fungi, why they know fungi? And then they learned, oh, the farmer just applied fungicide a week before without telling them. So it's very challenging to understand what is really happening, especially in the field.
Yes.
So for these things, maybe it's good to try it in smaller experiments, like in a pot or in a greenhouse first, to see what are the typical microorganisms that come with the plant, and then maybe give some idea of what might happen in the field. And of course, because variability is so high, it needs a lot of replicates, so a lot of repetitions, and you would have to study many years. So it's a matter of, it's at some point a matter of cost also. I think it's possible with enough samples to really get useful information, but it's very difficult to also control the conditions in a good way. And maybe one reason why soil is so diverse is just it has so many niches. There's so many pores, small particles, different carbon sources, there's insects, all kinds of things that happen at the same time. So there's so much variety in the environmental conditions that also then select for different microorganisms. So I think it's a very natural thing, but very challenging.
Because we are saying to recommendation to farmers, we are saying please use our biofertilizer, microbiology fertilizer. So after this using biofertilizer, the metagenomics all will change.
Yes.
And how will change this? This is very interesting.
Yeah, this is I think some behaviors are known. For example, the application of nitrogen or urea as an example. They have a different induce different changes in the microbes. What is also known, a colleague did this study, they had multiple generations. So they had canola, rapeseed, so the oil producing plant. They had 1 year on low nitrogen fertilizer and the next year on low fertilizer. And then they had the same on high fertilizer and next year on high fertilizer. And they also did the combination from low fertilizer 1 year, high fertilizer the other year. And what they observed was that those plants that were 2 times on low nitrogen fertilizer, they suddenly had nitrogen fixing bacteria in the seeds. So across the generations, the plant also remembers this condition by incorporating microorganisms that can be beneficial. And this is maybe a natural process. But of course it's very hard in the agricultural setting because after 1 year we harvest and we use the seeds to eat them. So this effect is also gone. And for bioinoculants, I think especially it's challenging to show how they establish. And what I saw from other projects, the most convincing for the farmers is, of course, if it changes the yield. But that's very hard because mineral fertilizers are so good that the plants just grow well. And the plants were selected to grow well on mineral fertilizer. So this is another thing that is often not considered. And what is useful in these cases is to have a control treatment where you reduce the fertilizer input. And then you can compare the addition of the biofertilizer if this increases. Because comparing against the mineral fertilizer or the conventional is very hard. It's, best case you keep the same yield or even lose some. But I think reducing the nutrient input and adding a biofertilizer is a good approach. Also to convince farmers, because you can say you need less fertilizer, but you apply this microbe and you get the same yield. Maybe not more yield, but the same yield. But you have to use less input. So this is another way of trying to frame the experiment. But I understand, it's very challenging.
Okay, how else?
Yes?
Thank you for the presentation. Very interesting. I have one question. Which database GenBank provides the most accurate bacterial identification until genus and also until species?
So this will depend on the length of your sequence. So if you have a 16S long read, then of course you can go more than genus.
For example, who 16S RNA.
Yes.
DNA.
Yes. So, for example, I mean, it's hard to say which database. I think NCBI is quite big. The NCBI is, I think, the most comprehensive one to date.
But we have a data gut microbiome analysis. We're working silver and green gene.
Okay.
But we have a lot of unclassified.
Yeah, I know the problem from
for
soil. so if you have a known database for gut samples, I think that is good if you have a specialized one, but I'm not aware of any. I think the most, silver relies also on the NCBI data, I think. Yeah, I think it's challenging. What we typically do with those that we cannot classify, we blast the sequence. We use blast. At least it gives you some hints that you can assign. But yeah.
Okay.
Yes, thank you very much
Yeah,
for
thank
your lecture. And I have a question. According to the metagenomics, there are usually short reads.
Yes.
We use an NGS, for example.
Yes.
And how it's enough to generate the whole length of 16S RNA or DNA or information about the speech.
Entire genomes.
Yes, yeah. Sure.
Yeah, I think this is the magic of the bioinformatic tools. There is some. It's not random. There's a lot of like smart people developing these algorithms. So there's different approaches based on the probability of which sequence follows and so on. But I'm also not expert in the exact algorithm. But as I mentioned, many of them also use a reference database. So they will first search for the closest resembling relative, and then they use this as a template to, for example, assemble the genome. Yeah, but it's true. There's no, at the end, there's no real way of knowing, and there's some other challenges. So the best approach would be, I think, to combine both short and long reads. Then you use something like PacBio or Nanopore for sequencing and short reads, because typically with the long reads you don't get the depth, but you get longer reads.
Yeah.
So, and with the short reads you get a high number, and so you can do good statistics, but you have, of course, less long reads. There are some algorithms that use both. Of course, it's twice as expensive, so I'm not sure it's always useful, but maybe for some cases it's useful if you want to really characterize one strain or something. But yeah, so this is, I think, it's challenging. I don't think there's an easy answer to this. Interestingly, it works quite well for the short reads, and many algorithms have been developed for this, so I think this is a balance also between how available are the tools, how good are the tools, to what is the biology behind it. No, it's a short read, so yeah. And this is especially challenging for fungal genomes because of the repeat sequences. Yeah. So this is, for fungal genomes, to assemble them, you always need a transcriptomics data as well to see if it's a functional gene or just an insert. So there is also, it's very tricky to find.
One more question, sorry. What kind of tools, I mean the omics tools, could be used for the separation of infected or non-infected healthy fruits or products? Do you know any cases for this?
Yeah, I think most of it can, for example, be done in China. Let's say first you have maybe amplicon sequencing, you just get which taxa is enriched in this fruit versus the other. And then you could do metagenomics as well to see what kind of functions they have. And if you do even transcriptomics, you can do differential expression between the two conditions. So you can see which genes are upregulated in the non-diseased fruit versus the diseased fruit. And the algorithms and tools are available in China, for example, the software.
Thank you very much.
Thank you very much.
Yes, yes. Yes, okay.